Okay, guys, a great night. Today, inshallah, we're gonna continue chapter 6, section 1. What does DNA look like, or how the DNA looks like? So, we said that scientists were interested to know the structure of the DNA, so they keep making researches and studies to be able to find what is the structure of the DNA. As we said in the last session, we know that the DNA is made of smaller units called nucleotides, and the nucleotide made of 5 carbon sugar, phosphate group, and nitrogen base, which could be A, T, C, or G. That's what we discovered later, but the question how science discovered that, so we passed by a history. Once a Chargaff, starting by the Chargaff rule, he analyzed the different samples of the DNA, and always he get the same result, that the amount of A equal T and C equal G, okay, and we call this the Chargaff rule, okay. Then we said Rosalind Franklin, brilliant woman, he worked by the X-ray diffraction technique, and she was able to find the specific details about the DNA, and but unfortunately her work passed to Watson and Crick by illegal way, as we explained last time, which pushes Watson and Crick one step forward. Okay, so they were able to publish their results or their research before Watson and Crick, so they got Nobel Prize about their model about the DNA. So today, inshallah, we're gonna continue. Okay, now we're gonna study the model of the double helix, or how the DNA actually look like. So students, okay, or physics students, you have an idea about this because we discussed that inside our classroom. But others, please pay attention with me. So Watson and Crick, they make their model, okay, based on the Chargaff's and Franklin's results, and they were able to build that model, which called the double helix model. So the model looked like a long twisted ladder. Do you know the rope ladder, guys, in which the two sides are made of ropes and there are wooden rings in the middle between these two sides? Do you know this? Like the one which is already used in helicopter and something like that. Do you know this kind of ladder, guys?
Yes,
Yeah. So
yes,
so here the two sides, the ropes which are fine, the two sides, it represent the 5 carbon sugar and the phosphate group, in which each nucleotide attached to the next, while the wooden rings in the middle, they represent the link between the nitrogen bases. So actually it's not that clear for us, so let's take a look to find it. Okay, so here we said that the double helix model, okay, so it's looked like that the DNA is made of two strands, two sides, okay, like the two ropes of the ladder, okay. These two sides are represent, or they represent the sugar and the phosphate, while the ranks in the middle, okay, they are represent the base pair. So that's how it's look like, guys. So here that the two sides that represent the group, okay, and that the ranks. So we are gonna find that each nucleotide is directly attached to the next. So that side just 5 carbon sugar attached to the phosphate of the next nucleotide, and then that sugar attached to the next phosphate of the next nucleotide, and so on like this. So they make like a skeleton, or here a backbone of the molecule in the two sides, while we find the nitrogenous base, okay, of each nucleotide is just fit to, or just bonded with the nitrogenous base of the nucleotide in the other strand. So you could see that they are complementary. That means the shape of A just fit with T and G fit with C. Could A, or could T fit with G, or A fit with C, guys? Can you see the shape could fit here, or that shape could fit here?
no,
No. So it means that the rule that always A is bonded to T and C bonded to G. It's a rule here that's based on the Chargaff results. Okay, so we call that the base pair rule. Base pair rule, that means always adenine is attached to thymine and cytosine is attached to guanine. So here that's what explain the results of Chargaff, that always he found the amount of adenine equal thymine and the amount of cytosine equal guanine, because they already attached to each other. Okay, so they are pairs, okay, like that. So here in that case we could find the bond between the nitrogenous base is represented by dots, while the bonds between the nucleotides here, or between the sugar and phosphate, they are represented by solid black line. Yes, guys, is it clear? So
yes,
which give a hint that this bond, the bond between the 5 carbon sugar and the phosphate, is stronger than the bond which attached the nucleotides to each other, or attached the nitrogen base to each other. So here this bond is covalent bond, which is stronger, harder to be broken, while the bond which attach the nitrogen base to each other is a hydrogen bond that easier to be broken. Okay, so is that good that the bond which hold the two strands be weaker than this? Yes, actually that's very important, and that's something which help in the work of the DNA. How? We know that before cell division, the cell should replicate. The DNA must be replicated. You get this, guys? We must double the amount of the DNA. Yes? Do you know this?
Yes. Yes.
Yeah, before the cell divide, it must replicate their chromosomes. That means must replicate their DNA to ensure that after division, each cell will receive complete amount of DNA. So here, that structure or this structure allow the cupping of the DNA. How to allow the cupping of the DNA? Because simply, okay, during division, the bonds between the nitrogen base will be broken, and the two strands will set apart, or they will be okay, just removed away from each other, and each side will be used as a template. Each side will be used as a template that used to build a new complementary strand. That means we're gonna use that code as a guide to build a new complementary strand. So if we find T, that means we're gonna put A in the other strand. C, we put G, A, T, G, T, and so on. And then same time, the other strand also build a new copy. So like this, we are ended by two new molecules of the DNA. Is it clear, guys? Okay. I'm gonna clarify that more for you. Okay. So here we said that the structure of the DNA, or how the DNA comes during replication, the DNA molecule is split down the middle where the bases met, and the bases on each side is gonna be used as a pattern or a template in which we are gonna make a new complementary strand. Okay, so here we're gonna find that the idea. Look with me. You can see this, guys. What happens? The DNA already was a helix. That means the nitrogen base were already connected to each other. Is it clear? Is it clear, guys?
Yes.
So
Yes.
before division,
Yes, miss.
we need to replicate the DNA. So what we're gonna do first, we must separate the two strands from each other. That means we're gonna break the hydrogen bond which is already attached the nitrogen base together. So we get two strands free. This the two blue strands. They are set apart. Yes or no? Is it clear? Okay.
Yes.
Then
Yes.
each side
Yes.
each side will be a template. Each side will be like a template. That means like a pattern. We are use it to build a new one. For example, if here C, so we're gonna put G. If there is A, so we're gonna add. Hey guys, if there is T, so we're gonna add A, and so on until we complete one new strand. And the same happen in the other side. So like this, after we complete the entire DNA, we are gonna have two molecules of DNA. Yes or no? Do you imagine this, guys?
Yes.
Yeah. So each molecule of DNA will made of two strands. One strand is already old, the one which is already found, which we used as a pattern or which we use it as a template, okay, to build a new strand. So that means each molecule of the DNA will have one old strand and one new strand, and that also will have one old strand and one new strand. So we call this semi-conservative model, okay? That means we just keep, okay, one strand old and we build a new one. Is it clear, guys, or there is something is not clear? If you can't understand, please ask. Don't just be silent. Guys, is it clear?
Yes.
Okay. So here, we gonna just make that more clear for you. Look at me carefully here, guys. Imagine that. Here we have the double helix shape, okay? So this dash represent the bond between nitrogen bases, okay? As we know that A must attach to T and C must attach to G. That's the base pair rule. So during replication, we're gonna break these bonds, okay? So we're gonna set them apart. So we're gonna find the DNA molecule will be look like this. They moved away from each other, okay? Like that. Is it clear, guys? So each side will be used as a template to build a new one, a new strand. For example, if the code here was A, T, T, C, G. So we're gonna build a new molecule, okay? Here, let's make it another color. So let's start. If that G, so what we should be here, what we should combine with G? Guys?
C.
C.
C. And here C, what it combine with?
G.
G. And T?
A.
A. And here T. Yes or no?
T. Yes.
So we start to build, and this will attach to each other. So, and then we'll keep doing this to the end of that. And the same side also we do the same at the opposite one, okay? Here we find if here that's A, so it will gonna T, and here that was sorry, that's T, so we're gonna here A, and that's here A, so we're gonna get T, and like that, that's here A, so we're gonna get T, and that's G, so we get C, and C we got G, like that. Okay, so we'll like this. We end after we finish. We have now two DNA molecule. That's one DNA molecule, and that's another one. So it's copied. Yes or no? Good idea?
Yes.
Yes. Each DNA now already has one old one, the red one, and one new, which is the purple. Yes or no? Good idea?
Yes.
After cell division, each cell will receive just one copy. That means this DNA will pass to a cell, while this will pass to another cell. Is it how like this, guys?
No.
No. So here like this, we find out that Watson and Crick was very accurate, that they was able to describe how the DNA looks like, and this model of double helix, and the idea of bonding between the nitrogen base by hydrogen bond, which is weaker than the covalent bond in the two sides, make it easier or make it good to explain how the DNA could be copied before the cell division. Any harder question here, guys? Any harder question?
No.
So, are you sure there is no question, guys? Everyone? Okay, so please, okay, we're supposed to study this lesson well from your textbook. Read it carefully, study it well, because it will be covered in the quiz next week. So every one of you, please start to cover the material or the 3 sections which we are studying. If you have any question, just write them down and try to contact me next session in the online session if you want to have any question, or in the classroom for the physical attendance student, to be ready for your quiz. Okay? Anyone need to ask about
Mr.
anything,
Ahmed,
guys?
I have a question.
Yes.
Yes, me.
Yes, Farah.
Mr. هو chapter 6 section 1 the worksheet of it is posted on SMS or no?
No, not yet. Okay, so it will be posted inshallah today.
Okay.
Okay.
Thank you.
Well, thank you. Goodbye.